Hello everyone, we're talking about protein synthesis today, so how we take our DNA code and use it to create the proteins that become our enzymes and our structure and basically do everything we need in the cell. Here are your key terms, and let's start off with a couple basic definitions here. So DNA makes up our genetic code. You've probably heard that before. And if you take all of the DNA in a cell or an organism, we call that our genome, the totality of all of our DNA code. Now, the code itself is based off of the order of the bases. So whether you have an A or a T or a C or a G and what order they come in, that is the code we're talking about. So that code is what's going to tell us what protein to make, how to make it, when to make it, how many times to make it. Basically, it tells us, it's the blueprint that tells our cells what traits to express. Now, these proteins will help form our structure and our enzymes, and they will then help dictate the function of the cell. We actually need these proteins and enzymes to be able to do a lot of the copying of our DNA and to do protein synthesis, so they kind of work hand in hand with each other. Now, sorry about that. Now, genes are the portion of our genome that codes for a specific protein. So not all of our genome is actually made of genes. There's only about 20 to 25,000 genes in the entire human genome. So not as many as you might think. Only specific portions code for a protein, and that's what is a gene. Now, the rest of the parts that don't code for a protein, it's not that they're not important. They're actually still very important. These are often our regulatory sequences, so they will tell our cells when to make the gene turn into the protein, how often to turn it into a protein, how many copies to make, when to stop making copies of it. It's the regulatory portion. And even still, there are some parts of our DNA that we often call junk DNA because we don't really know what it does, but we definitely know it's not junk. It's important. We just haven't figured out what it's used for yet. So gene expression or protein synthesis, they're basically the same term. This is the process by which we take our genes, transcribe them into RNA, and then use that RNA and translate it into a protein. So it's basically taking the DNA, the gene, which is the blueprint, and getting some builder who can understand the blueprint and build the structure, the protein from that blueprint. Now, all of this brings us to our central dogma. This is basically the core tenet of biology that says the flow of genetic information goes from DNA to RNA to a protein. This does not happen backwards. So we can't take a protein and turn it into DNA. The DNA is what codes for the protein. Now, there is one exception, and that is retroviruses, something like HIV. It can't go from a protein back to DNA, but it can go from RNA and convert back and forth between DNA and RNA. So it can go backwards only in this direction, not from the protein direction. So there's two steps to protein synthesis or gene expression: transcription and translation. Transcription is when we convert the DNA code to RNA, in particular mRNA. And this is going to happen in the nucleus because that's where we find our DNA. DNA doesn't leave the nucleus in a eukaryote. Then translation is taking that RNA code that we just copied and converting it into amino acids and creating the protein from those amino acids. And this is going to occur in the cytoplasm where we find our ribosomes. So before we get into those two parts, we have to understand the three types of RNA in a little bit more detail. So the first is messenger RNA or mRNA. This is going to be a single strand, and it can leave the nucleus and go interact with a ribosome. This is going to be the copy of the DNA. So when we take our gene and we copy it into RNA, this is the RNA that we copy it into. So it's literally going to hold the DNA code. Then we have transfer RNA or tRNA. This is a molecule that is twisted into a T shape, and on one end it has what we call an anticodon. I'll explain what that is more later. And the other end is going to hold an amino acid. So this is going to be important when we're building our protein at the ribosome during translation. And then we have ribosomal RNA, rRNA. Now, ribosomes are just RNA and protein working together. So it's just one part of that ribosome. It's the part that's going to actually make the reaction happen between the amino acids. It's going to connect them together and form that peptide bond that connects amino acids together. So let's start with transcription. This is how we go from our DNA code and we copy it into mRNA. If you look at the word transcription, this is the word like transcribe. This is what you're doing when you're taking notes. You're seeing something or hearing something in the same language and you're copying it down in a different format. So while I am giving you the auditory English, you might be copying it down into written English. It's still the same language. DNA and RNA are both nucleic acids. You can consider them as speaking the same language. So we're just transcribing it. We're just copying it. It's just in a slightly different form, but it's saying the same thing. So the ribosomes that make the proteins, they're found in the cytoplasm, but DNA is found in the nucleus. So we need a way to get the DNA message out of the nucleus and to the ribosome. That is the whole purpose of transcription. So what we're going to do is use an enzyme called RNA polymerase. If you remember when we talked about DNA replication, DNA polymerase used the DNA template to make a new strand of DNA. It copied the DNA. RNA polymerase is going to do the same thing. It's going to read the DNA and create RNA to match the DNA template. In this case, it's going to make mRNA, the messenger RNA. So that messenger RNA is going to be able to then leave the nucleus and interact with a ribosome. So it's copying the gene, the important blueprint of our DNA, leaving the nucleus and going to the place where we can read that code and turn it into the protein. So RNA polymerase is going to first attach to the DNA at a specific site called the promoter. This is one of those non-coding parts of DNA, that junk DNA that regulates how we work, how we read our genes. So this particular site, the promoter, is telling the polymerase, hey, a gene is about to begin after me, so attach here and you'll be able to copy the whole gene. And from there, it moves, same thing as DNA polymerase, in the 5' to 3' direction, and it follows the same base pairing rules. C bonds with G, and A bonds with U. When we copy DNA, remember A and T go together, but because we're making RNA, RNA doesn't have a T, it has a U instead. So the A would bond with a U whenever you would want to put a T. So if we had a DNA template that said TAC, TAG, AGCA, you would then follow base pairing rules to figure out the RNA strand. So what bonds with T? Well, that's A. What bonds with A? Normally you'd want to say T because that's what bonds in DNA, but there is no T in RNA, so instead we put a U. C bonds with G, T with A with U again, and so on and so forth. So that would be how you would do an example problem of transcription, taking that DNA template and converting it into a strand of mRNA. Now, translation is a little bit more tricky, a little bit more complicated. So DNA and RNA are both nucleic acids, but proteins are a completely different type of biomolecule. Nucleic acids have the nucleotide as their monomer. Remember, the monomer is the building block. Whereas proteins, their building block is amino acids. So now it's like we're speaking two chemical languages, two different languages. So we're going to have to translate the RNA into protein language. Now, when you translate, if you saw the word hello, you wouldn't translate it by saying H, okay, what does that translate to? E, what does that translate to? You would translate the whole word. And the same thing is going to happen when we talk about translation in the cell. You're not going to translate it letter by letter. We're going to end up translating it word by word. So what do I mean by that? Well, what is a DNA word and what is a protein word? Sorry about that, guys. A DNA or RNA word is called a codon. So this is a sequence of 3 bases that are right next to each other, 3 mRNA bases that are going to end up being able to be translated into a single amino acid. So the codon is the DNA word. So here, if this is our mRNA, AUG, AUC, UCG, UAA, we can take the first 3 letters, AUG, and that now becomes a word. I can then use a dictionary to translate and say AUG means met in amino acid or methionine. So that's how we are able to do this translation, because we group together 3 nucleotides, 3 bases of mRNA. Now we end up using a chart. There is a wheel and there's also a square chart that I'm going to show you. And this is going to help us convert those codons into amino acids. So in the middle we have the purple, the gray, and the blue. Those are our codons, and on the outside in white, red, and green, we have our amino acids that they match up to. Now, this same chart is used to decode between RNA and protein, whether you are a human, a bacteria, a dolphin, a penguin, whatever it might be. The whole code is universal. So every single organism on Earth uses the same code. That points to the fact that we all evolved from the same organism that evolved to use this as their translating code. Now, if we look at this too, we can also see that CGU, CGC, CGA, and CGG all give us the same amino acid, arginine. That is a redundancy. Multiple codons can end up coding for the same amino acid. I want you guys to think about why that might be important and why that might be useful to a cell, and we'll talk about that in class together. So in this case, another example here, all 4 of these code for the same amino acid, proline, and we call that redundancy. So this code is both universal, meaning all organisms use it, and it has redundancies in it, where sometimes multiple codons can give you the same amino acid. So let's talk about how you read this codon wheel now. You're going to read it from the inside going out. And we separate it into 4 quadrants for the 4 different bases. So the first way to look at it is you have the vertical line here and the horizontal line here that separate it into a G section, a U section, a C section, and an A section. So the 4 different quadrants. And it all depends on which base is first in your code. So in this case, we have the codon AUG. The first letter is A. So when I look, I'm going to start in the middle with the large purple letters, and my first letter is going to correspond to A. That now tells me I'm somewhere in this lower left quadrant. Now, the gray in that quadrant represents the second letter in my codon. So in this case, that second letter is U. So now I'm going to say, okay, I had A. Now I'm going out to the U in that quadrant. So now it's narrowed it down to just this area right here. It's going to be one of these 2 amino acids in this area. I have discounted or I have eliminated, that's the word I'm looking for, all of these three other options in the quadrant because I know my second letter is a U. Now, your final letter is going to be in this blue ring. So we're in the bottom left quadrant. I found out that the first letter was A, the second letter was U. Now here within this tiny area that corresponds to the U, I'm going to look for my third letter, which was a G. So I have U, C, A, here's the G. So that's telling me AUG gives me the amino acid met or methionine. So you're always going to work your way out from the purple first letter to the gray second letter to the blue third letter, and it will tell you what the amino acid is. Now these are all abbreviations for the amino acids. They have full names. We just shorten them to the first three letters. You will not have to know their full names. You will be given this chart and you'll just have to write down the abbreviation. So one more example here. If we have, let's do UCG. So I start in the middle with my first letter, which was a U. So now I know I'm in the U quadrant, the upper right quadrant here. So I've eliminated all of the rest of this wheel. My second letter is a C. So now I'm going to look for the gray in the second or in the upper right quadrant. So U, C. So now I know I'm just going to be in this area, and sometimes you can skip ahead. It doesn't matter what my last letter is because UC whatever gives me serine. But just in case it's UCG, so UC go out to the last letter G, and that gives you serine. So when you have a case where there's multiple letters and they all just give you the same one, that means any one of those options, UCU, UCC, UCA, UCG, they all give you the same amino acid. That's where that redundancy comes in. So as I said, there is the wheel that we can use, but there's also this chart. I will give you both on the quiz. The chart, in my opinion, is a little bit easier to understand. So you have on the left-hand side here your first letter. Your second letter is up on the top, and your third letter is on the side. So let's go with this first one here, AUG. So my first letter is A, that means I'm going to be in this row. My second letter is a U, that means I've narrowed it down to this box right here. My third letter is a G, so I know in this box I am AUG right here, it's met. Or you can just narrow it down first, second letter, and then just look through here to see what your particular codon is. So both will be available. However you learned or whatever you prefer, you'll be able to use. So with translation, the mRNA is going to join up with the ribosome. The ribosome is this green portion here. It's actually two pieces that come and clamp down on top of the mRNA. And then a tRNA is going to come in and carry the amino acid that matches the codon. And from there, the ribosome is going to, the rRNA part of the ribosome is going to catalyze that formation of the peptide bond and cause those amino acids to stick together, and it's going to grow the polypeptide chain. This is the primary structure of the protein. Remember, primary structure was just the order of the amino acids. These are now going to either form those alpha helices or the beta pleated sheets, and that secondary structure. Then they might twist and turn on each other more to form their tertiary structure, the active structure. Then one protein can join with another protein to make quaternary. So we are talking about forming that primary structure, the order of the amino acids based on the DNA code. So the tRNA, this is the RNA right here, you can see that it turns on itself in self-base pairs, and it kind of forms a T-like structure. On one end, we have what's called an anticodon, an anticodon. This is going to have the opposite code of the codon for the mRNA. So it's going to be able to base pair to the codon. That's how we know, that's how the cell knows what codon corresponds to what amino acid. So the anticodon is going to carry the amino acid that matches the mRNA codon. So what happens is As the mRNA is in the ribosome, these tRNAs come in, they base pair to the mRNA, and they deliver the correct amino acid. Now, when we talked about transcription, we saw that the RNA polymerase knew where to attach to the DNA because it found that promoter sequence. That promoter sequence said, hey, attach here, a gene is going to come very soon. So how does the ribosome know where to start? Where does that protein actually begin? Well, the ribosome is going to attach and bring the first amino acid in at one very specific spot. It's going to start at the start codon, and the start codon is always AUG. So this also sets the frame of how to read the DNA and the mRNA. So it says the gene starts here. It doesn't start at U, it doesn't start at whatever comes before the A, it doesn't start at G, it starts with AUG, and it sets the tone and the groups of 3 letters from that point on. So that means that every single protein is going to start with the same amino acid. It starts with methionine because that corresponds to the start codon. So then how does the ribosome know how to stop translation? Where does it know the end of the gene? Well, that's when it reaches a stop codon. There are 3 stop codons in the entire code. It's UAA, UAG, and UGA. All of those correspond to what's called a stop codon. Now, these do not have amino acids that correspond to them, so the tRNA will basically be empty. And when the ribosome tries to create a peptide bond, it will fail. And when that happens, the ribosome will break apart from the mRNA and the whole process stops. So we start with the start codon, AUG, that corresponds to the amino acid methionine, and we stop when we reach one of the 3 stop codons, and there's no amino acid that corresponds to those. So we started with our DNA code, we copied it through a process called transcription. We used RNA polymerase. It read the DNA code and through base pairing rules added the opposite pair, so T with A with U, and C with G. And then a ribosome translated that mRNA into amino acids, and the ribosome connected those amino acids together to form the primary structure of the protein. So that is it for this particular topic. There's one more topic about mutations, and that will be it for today.